Welcome everyone to the first Tuesday of March. So we have a great speaker today. I just want to bring your attention a little bit to some of the upcoming events. We have a neuroprosthesis seminar. I keep saying we'll fill those in. I haven't done that yet, but we will. Monica Perez will be here in May, or not here, but live on your computer screen in May. We have, is it going to rotate here to, we have other first Tuesdays, and we have our other activity, which is Behind the Science right here. And this is basically describes what kind of resources that the FES Center can provide and figure out what we need to provide. Okay, so that's monthly as well. Yeah, so the first Tuesdays in April, we're going to have Asif Sheikh and in his group, and there's an available spot on May 4th. If you want to talk about your work in May, please let us know. Okay, so why don't we get started? So our speaker today is Andrew Pieper. He holds about, I don't know, 25 different positions, it looks like, around town, but he is the director of the Neurotherapeutic Discovery of Neurotherapeutic Discovery in the Harrington Institute of University Hospitals. He's an investigator in the Harrington Discovery Institute. He's a psychiatrist at the Cleveland VA Medical Center. He's the Morley Mather Chair in Neuropsychiatry at University Hospitals, among others. So he has his PhD and MD from Johns Hopkins University. And Dr. Pieper does a lot of, like it said in his title and in my introduction, he does a lot of therapeutic development for neuro disorders, especially neurodegenerative disorders. So Andrew, I think I'll let you describe your work instead of me trying to do it. So thank you for joining us today.
Oh, well, thank you. Thanks for the introduction, and thanks for the opportunity to present to the FES Center. Let me see if I can properly share my screen. Okay. Oops, hold on a second. Does everybody see the one slide in presentation mode?
Oh. We see your desktop.
Which
looks like mine, by the way.
I have three desktops. Hold on.
We're looking at the mountains, if it helps.
Yeah.
Okay, hold on a second. It's weird. You know, Eric, it took me through this and it all worked fine.
We're looking at the probably your main desktop that has all of your icons on it. If you go to share screen, it usually gives you a choice of which screen to share. Sometimes if you just click the PowerPoint, now it's just a blank desktop.
Yeah, I think this should work.
You probably picked like screen 2 or 3.
Oh, yep. There we go. Okay, now we're set.
All right, so now I have to get all of it. Well, okay. It's not necessarily gone so smooth here as I've shown you all the slides in reverse, but let me get back to the beginning. Okay.
Everyone avert your eyes.
All right.
There we go. There we go.
Used to present with a slide carousel with actual real slides. It was actually simpler than I guess for me, but okay. So, all right. Well, anyway, as you can see, as the title says, I'm going to talk about work that we're doing in our lab to develop new pharmacologic approaches that we hope will enhance functional recovery after brain injury. And I want to start with just this slide that shows the magnitude of the problem, which is that deterioration and death of nerve cells in the brain is afflicting about 50 million people worldwide right now. And if we don't find something that stops it, it's projected to triple in the next 30 years. In addition to the massive suffering that this entails, the cost of caring for these patients is going to exceed over $1 trillion a year in the United States alone by 2050. So unfortunately, as most of the people here know, despite the magnitude of this problem, we still don't have any medicines that can stop cell death in the brain. And I think that's because the brain is one of, if not the most complicated organs in the body. And as a result, the field of brain health is about 10 to 15 years behind the other fields of medicine. For example, even though Alzheimer's was discovered 120 years ago, there have still only ever been 5 medicines approved for treatment of Alzheimer's disease. And those only address the symptoms. None of them actually stop the nerve cell death that goes on and causes progression of the condition itself. And I think that's primarily because we just understand so little about the process of nerve cell death. So right now, the field of medicine is such that we have lots of great medicines for the other organ systems of the body that are helping us live a lot longer, but we aren't necessarily living any better because most of us are getting older and acquiring these currently untreatable neurodegenerative conditions. So with that in mind, in our lab, we took a non-traditional approach to try to help attack this problem. We figured that because we simply didn't know how to recapitulate inside of a test tube the complicated nervous system in the context of the entire brain, we decided instead to use the brain itself to identify new drug-like molecules that would help nerve cells live longer. And our straightforward goal was simply to go into this, not worrying about the mechanism by which it might work, but simply to try to find a small drug-like molecule that would work safely in the brain, regardless of how that mechanism might be. So to do this, we delivered thousands of small molecules directly into the cerebrospinal fluid circulatory system of the brains of mice in order to see whether any of those molecules could make nerve cells survive in the region of the brain known as the hippocampus. And we focused on that region for a few reasons. First, it's a critical part of the brain that controls our emotions and our ability to learn and remember new things. Second, it's highly sensitive to injury and disease. And in fact, in most neurodegenerative conditions, as well as most chronic psychiatric conditions, it's the part of the brain that's affected earliest and most severely. And then the third reason was one of practicality. The hippocampus is one of a few regions of the brain that makes new nerve cells every day throughout our entire lives. And this is thought to enable us to learn and adapt to a changing day-to-day experiences. And we actually make about 10 to 20 times more nerve cells every single day than we need. And so as a result, the majority of them die. And so we decided to use this as sort of like the test tube and simply look to see if we could find any molecules that would make less of those nerve cells die, make them survive at a higher rate. And so essentially we made our brain into the test tube. And after a long process, we discovered that this compound shown here doubled the survival rates of those nerve cells. I named this compound P7C3 because it was compound number 3 from pool number 7. And shown below in red is an example of the greater number of neurons that survive in the hippocampus of an animal that's been treated with P7C3 compared to one that received just the vehicle alone. And so importantly, not only do we find that it worked very well in rats and mice, but we also found that it works very well in monkeys. And so here is an example of what we see in the brains of rhesus monkeys that have been fed one of our most potent analogs of P7C3, known as P7C3A20. This graph here, the first graph in the middle, shows the increased number of surviving neurons in the hippocampus of the monkeys that consumed P7C3 every day for 9 months, relative to those that ingested only the vehicle. And then on the right are examples of the staining of surviving nerve cells under the microscope. The arrows are pointing to them. It might be hard to see on the screen, but what you can see is that there are many more that survived in the animals that consumed the P7C3. So ingestion of P7C3, oral ingestion of P7C3 in a primate promoted survival of young hippocampal neurons. So over the past several years, we've expanded our knowledge of how P7C3 works to protect nerve cells. Again, remember, we found it in what I call a target agnostic way. And we now know that it acts directly to preserve the cell's ability to manufacture energy in times of stress. So an analogy I like to use is that just like a car uses more gas to drive up a hill or to go faster, cells need energy when they have to work harder. And I want to remind everyone here that our nerve cells actually have lots of ways to repair themselves in injury or disease, but these processes require energy. And when energy demand becomes too great, such as in times of overwhelming injury or catastrophic onset of disease, the cell runs out of energy and dies, just like the car dies when it runs out of gas. But the difference is once the cell is dead, you can't go back and fill it up with more energy. And so what we can do to help the cells, the nerve cells, is to keep them from ever losing their energy in the first place. And that is how we have learned that P7C3 works. It helps cells preserve their levels of an energy metabolite known as NAD. So to date, both we and others around the world have applied P7C3 to various animal models of neurodegeneration, many of which are shown on this slide. We've learned that P7C3 works not only to protect young newborn neurons in the hippocampus, but also to protect mature nerve cells elsewhere in the brain, the spinal cord, and even the peripheral nervous system. So protective activity has been reported in animal models of traumatic brain injury, Parkinson's disease, Alzheimer's disease, stroke, peripheral neuropathy, spinal cord trauma, ALS, and even depression, anxiety, and post-traumatic stress disorder, which I think a lot of people don't realize are associated with neurodegeneration, especially in the hippocampus. And so now that we know how it works and we've made improved derivatives and there seem to be potentially many helpful applications for this if it were to become a drug, we're working very closely with a drug development team at the Harrington Discovery Institute to try to make P7C3 into a medicine for patients by further optimizing its chemical properties and implementing other key steps in that process. At the same time, however, in the laboratory, we're using P7C3 as a tool compound to learn new things about the biology that surrounds nerve cell death. And at the moment, we are primarily pursuing this through studies of traumatic brain injury in mice. So abbreviated as TBI, this condition is one of the most serious afflictions that affects society today. TBI is caused by anything that hits the head. It can happen at any age. For example, in young people, it's common with contact sports. In the military, it's common with explosions. And in the elderly, it's very common with falls. The worldwide prevalence of TBI is enormous. And in the US alone, it affects more than 3.5 million people a year. I also want to note that TBI is the biggest non-genetic, non-aging related risk factor for Alzheimer's disease. And basically, I think it's safe to say that TBI is probably the biggest underlying cause of neurodegeneration today, but no one really knows why. We don't understand what happens after a blow to the head. Why does that start a cascade that causes, in many cases, a lifelong chronic process of neurodegeneration? So consequently, we have no way to protect ourselves after this injury occurs. And so we've been studying this problem in our lab. TBI in people is caused by many different ways, and 3 of the principal forms are shown here at the top of the slide. There can be a direct concussive impact. There can be acceleration, deceleration injury as the head is rapidly moved and the brain kind of sloshes against the sides of the skull. And there's also a phenomenon of shockwave injury where a blast wave passes through the organs, including the brain. We in our lab have developed an apparatus that incorporates aspects of these 3 conditions. And our TBI machine is shown here on the bottom of the slide. It's about 10 feet long. And as you can see, it looks like a small yellow submarine. It consists of 2 chambers that swing apart as shown here in these pictures. And the mouse goes in the left chamber. And then a mylar membrane is placed in the opening of the right chamber. And the apparatus is then joined back together. And we increase the pressure in the right-hand chamber until the mylar membrane pops. And when that happens, it creates a sudden jet of air that causes a global concussive injury to the mouse's head, as well as an acceleration, deceleration as the head is moved very rapidly. And then there's also a component of early blast wave exposure. So I just want to say that, in my opinion, there is no perfect model of TBI in animals because there's no single form of TBI that people endure. There's weaknesses and strengths to all of them. This is the one that we use. We like it because it causes a very reproducible chronic form of injury that begins with axon degeneration and progresses over time to nerve cell death. And so it offers us a lot of opportunities to dissect out different pathways that lead to nerve cell deterioration after traumatic brain injury. So to begin, when we first applied P7C3 to the model, we noted that if we waited 24 hours after injury and then began administering the compound, we fully protected mice from developing memory deficits, from developing axon degeneration, and also from developing motor problems. And this was a very exciting finding because there are certainly lots of people every day experiencing a TBI who could conceivably get treatment within 24 hours. We waited 24 hours because we figured most people could probably get care within that time. And so this is very inspiring. But that's not the case for everyone. And there are millions of people worldwide who have suffered a TBI in the past, including decades ago. And most of these people are experiencing these chronically progressive neurodegenerative worsening conditions. And the dogma in the field has long been that there's nothing that can be done to stop that chronic neurodegeneration, and that the best treatment that we can give these patients is a supportive treatment with a combination of rehabilitative therapy and medicines for symptoms, which help, but don't stop the chronic neurodegenerative process. And so we decided next to see, could we use P7C3 to address this other aspect of traumatic brain injury as well? And this is how we did it. We gave mice the same form of traumatic brain injury when they were 2 months old, and then we just put them back in their cages and we let them live there normally for an entire year. So mice normally live 2 to 3 years. So 1 year in their cage is like the equivalent of decades in a person. So then 1 year after the injury, we then started to treat mice with either vehicle or with our P7C3 A20. And when the mice were 15 months old, meaning we treated them every day for 1 month, we tested their memory in Morris water maze. And remarkably, the mice that were treated with P7C3 showed fully restored memory, identical to the mice that had never been injured. Memory is shown here on this graph as the number of platform crossings in the probe test when the escape platform has been removed. It reflects how well the mouse remembers where it previously learned the platform to be. So this was exciting in itself that we were able to restore memory function at such a chronic time point after the injury. We actually had no idea if this would work or not. So we were excited that it worked. And the next thing we wanted to know is, had we really fixed them, or would the deficits return? And so we stopped treating them and put them back in their cages for 4 more months, and then put them back into another water maze with the cues moved around so they didn't remember it from the last time, and tested them again. And we saw that the protective effect persisted. While with aging, they all showed an expected decline. You can still see that the P7C3 treated animals performed as well as the sham injury animals. And so we were kind of blown away that we had actually caused some sort of permanent change by treating them at this chronic time point. It would not have been what we would have hypothesized. So we then looked at the brains under the microscope, and we discovered that P7C3 had actually stopped all axon degeneration in the injured animals. And the way that we measure that is through silver staining. And if you look in the middle panel here with the TBI vehicle, you see a lot of black degenerating axons in the animals that received vehicle and had the injury. But there's a relative lack of that staining in the sham injury animals or in the animals that received TBI. So we then looked further under electron microscopy and think that we may have figured out why this was the case. So here you can see that P7C3 treatment repaired the brain endothelial cells, which normally form a tight blood-brain barrier at the junction of the vascular system in the brain. The function of that is to protect the brain from non-specific entry of things into the blood. And the red arrows here in the middle panel point to breaks in the endothelial cells that developed 19 months after the animals had experienced a TBI. And in the animals that received P7C3, 4 months prior, actually just for 1 month, they had no breaks. That we had helped, the compound had helped endothelial cells repair themselves. And we then looked at this further. I'm not going to show it here. It's all published, but we saw that this was achieved in part by helping elevate levels of tight junction proteins known as claudin 5 and zona occludens, which help the endothelial cells join back together. And then we also showed that P7C3 can protect human brain microvascular endothelial cells in culture as well. And basically, so I'm showing this as an example of how we used P7C3 to identify deterioration of the blood-brain barrier as one possible contributing factor to the chronic lifelong neurodegeneration that happens after TBI. And it actually, I believe, changes the outlook of the field by showing for the first time that it's possible indeed to pharmacologically reverse the damage that has occurred after brain injury long in the past, which I don't think many people would have thought was possible before. So that's one example of how we use P7C3 to discover new biology about neurodegeneration. And now I want to show just one other example, because I know we're just supposed to have a short talk today. In wondering how axon degeneration after TBI might be linked to increased risk of Alzheimer's disease, we turned our focus to the tau protein, which has long been known to be involved in Alzheimer's. And its normal function is to control stability of axons. So axons, as you all know, are the long processes that nerve cells extend to communicate with each other. And they're extremely energetically demanding, extremely dynamic, constantly reforming themselves and such. And tau plays a very important role in that. And it's been known for a while that the tau protein gets modified in Alzheimer's disease by a process called acetylation, as shown here, which is simply the addition of acetyl groups to tau. And so we wondered if this might also happen in TBI. And we observed indeed that tau is acetylated pretty rapidly after TBI, as shown here by Western blot for with antibodies that recognize the acetylated group on the tau protein. And then we showed that the specificity of this effect was due to the ability of P7C3 to elevate NAD and drive sirtuin-mediated deacetylation. And so if you look at the diagram on the bottom, when we gave either FK866, or which shuts down the NAMPT enzyme, or when we gave EX527, which blocks sirtuin, we prevented P7C3 from being able to lower the acetylation of tau. And so that told us that this process of regulation of acetylation of tau after TBI occurs, first of all, which nobody had shown before, and then we wondered what might it be doing, because normally tau is restricted to the axon and doesn't go into the cell body. But when we looked under the microscope, we saw something very surprising, which is that two things happened. One, shown here on the left, the axon initial segment itself degraded. And so the axon initial segment is this tight meshwork of proteins at the junction of the axon and the cell body that prevents or regulates the flow of things between the cell body and the axon. And normally, one of the things it does is it restricts tau out into the axon. After tau acetylation, the axon initial segment breaks down, which you can see shown here with loss of staining of two of the key proteins that make up this component. And as a result, tau, which in the top panel shown on the right, should normally be in the axons, in these long axons, it then travels into the cell body where it shouldn't be. And so it pathologically invades from the axon into the somatodendritic compartment, and ultimately then leads to neurodegeneration, axon degeneration, nerve cell death, and acquisition of all kinds of major nerve behavioral deficits. So the entire process is, again, this whole process is blocked by treatment with P7C3, and which you can see in the middle panel. And then the bottom two rows are the same pharmacologic blockers that prove the specificity of P7C3. And basically, to summarize a very long story, this paper was just accepted last week, and this is our graphical abstract of what we have uncovered. There's this new pathway in the brain after traumatic brain injury that leads to neurodegeneration that begins with modification of the GAPDH protein by S-nitrosyllation. Once GAPDH is S-nitrosylated, it coordinately activates the P300-CBP enzyme to add acetyl groups to the tau protein, and it simultaneously inhibits the sirtuin-1 enzyme, which removes those acetyl groups. So you get kind of a double whammy on the tau protein. You increase acetylation and you decrease the removal of the acetyl groups. And this net increase in tau acetylation leads to breakdown of the axon initial segment and tau mislocalization out of the axon and into the cell body, which causes neurodegeneration. And remarkably, we found that acetylated tau can also be detected in the blood. People have been trying to use tau in the blood as a marker for a lot of things with some limited success, because what's weird about tau is that it's so small that it just freely diffuses between the brain and the blood. So it actually accumulates in the blood relative to things that are going on in the brain. But when people have tried to look, for example, at phosphorylation, they don't see as robust of a change. But as luck would have it, the acetylation is not changed between the blood and the brain. And we actually have discovered that this is a very accessible way to ascertain the degree of neurodegeneration in the brain. Right here, we see a very tight correlation between the magnitude of tau acetylation in the brain and the magnitude of tau acetylation in the blood. And so we then wondered if this is happening in mice, might it also be happening in people? And so we teamed up with a group of neurologists at UT Houston to examine the blood of patients who had suffered from a TBI 24 hours prior. And we saw indeed that the same effect held true. After a TBI, the levels of acetylated tau significantly increase in the blood of people, even while total tau levels remain the same. And so I want to close with just one more piece of data that we're pretty excited about here. If you direct your attention again to this diagram, there are 3 points shown in purple where we use a therapeutic agent or a genetic modification to interrupt neurodegeneration after TBI in our mouse model. First, at the top, we blocked S-nitrosylation of GAPDH after TBI with a compound called omigapil. Then downstream, we also achieved protective effect by elevating NAD levels, which increased activity of the deacetylase enzyme sirtuin-1, thereby lowering the amount of acetylated tau. And then lastly, all the way on the far left, we showed that treatment with either salsalate or diflunisal both protected animals from TBI as well through the same process by preventing acetylation of tau. And what's unique about these two drugs is that they're both NSAIDs, non-steroidal anti-inflammatory drugs that have been used for their anti-inflammatory properties. But these two NSAIDs, unlike all the rest of the other NSAIDs, also have this additional property of inhibiting the P300-CBP acetyltransferase enzyme. So aspirin doesn't do this, naproxen doesn't do it, ibuprofen doesn't do it, but salsalate and diflunisal do it. And so we calibrated down to levels of these NSAIDs that were not anti-inflammatory and showed that they still achieved a protective effect by inhibiting the P300-CBP acetyltransferase. And so we wondered more broadly, might these be associated with decreased incidence of neurodegenerative disease in people? And so to do this, we teamed up with Dr. Feixing Cheng at the Cleveland Clinic, who has access to a market scan database of over 7.5 million, actually now it's 200 million. The time we did it with him, it was 7.5 million patient prescribing records and diagnoses. And just ask the question, when compared to aspirin, are salsalate or diflunisal associated with a decreased incidence of developing Alzheimer's disease? And they both are. And so you can see here that over a 6-year period, patients who received either salsalate on the left or diflunisal on the right had a significantly decreased rate of developing Alzheimer's disease. And the fact that diflunisal is appearing to be more protective than salsalate was very encouraging for us, because diflunisal is also much, much more potent of an inhibitor of the P300-CBP acetyltransferase enzyme than diflunisal is. So we think it's possible that there already are some FDA-approved drugs that could go into clinical trials for neuroprotection that might have an unexpected effect that we didn't realize before. So in conclusion, we implemented a target agnostic in vivo screen in mice that identified P7C3 as an NAD enhancing small molecule with neuroprotective efficacy across multiple animal models. In addition to, hopefully, we hope very much that it's going to form the basis for a new class of neuroprotective drugs. We've also used this discovery as a tool compound to challenge the dogma of how chronic TBI could be treated, to identify blood-brain barrier as a potential therapeutic target in chronic TBI, to, I believe, potentially have unearthed one of the links between TBI and the increased risk of Alzheimer's disease. They both involve degradation of the blood-brain barrier. It's entirely possible that this could be something that links the two together. And then to identify already FDA-approved drugs that might have some unanticipated therapeutic effect in neurodegenerative disease. And then I would like to close by thanking the people in my lab, all of whom shown here were involved in this work, especially Min-Kyu Shin and Edwin Vasquez Rosa, who are co-first authors on the acetylated tau paper that I just told you about.
Oh, thank you, Andrew. That's fascinating stuff. That's really fascinating. I have to say, my first inclination was, why don't we just put the P7C3 in like breakfast cereal or something and let everybody eat it every day? You know, are there side effects?
We haven't seen any. we haven't seen any side effects. I know it sounds strange, but we haven't. We've given it to mice, to Alzheimer's models of mice for over a year every day. They didn't have any side effects. We gave it to monkeys orally at UC Davis. We didn't do it. The people at UC Davis did it. They gave it to them orally every day for 9 months. And then we sent all of their organs to pathology contract research service and said, find something that went wrong. And there was no damage anywhere. So
Wow.
I'm sure that's what we're doing with the Harrington Discovery Institute. I'm sure that there will be things in the structure of our lead molecule that are not ideal for drug development, and that it's going to need to be tweaked somewhat. But there isn't anything massively overt that we've seen. And I kind of think that maybe, well, one, we got really lucky, but two, I think that's sort of the strength of the original in vivo screening approach that we did. Was if it had been really something that made them sick in the first place, it never would have come out as an agent.
Well, I have a few more questions, but I'll open it up to others that would like to ask.
I'll stop sharing my screen so we can all see each other.
So questions for Dr. Pieper? Well, I have to say that, you changed my ideas about some of these disorders, because it seems like it's a not so much a neurological disorder as it's the support cells, the endothelial cells, and things that some of your, well, what you presented, it seemed like it was protecting, the endothelial layers or regenerating them, and not necessarily the neurons, right?
Right. One thing that we think is going on is that by repairing the blood-brain barrier, we're helping the brain equilibrate and repair itself. One thing that we don't understand, I don't think anybody understands, is why does the blood-brain barrier deteriorate chronically after TBI? So it makes sense that it would be disrupted first because there's a force, and we see that. But then we see that it repairs itself. And so and we're currently charting that out now in great detail. But what we've seen is that so far is that very quickly it opens up really wide, stays open, and then closes back up, and it's closed for a long time. And then chronically it starts to open up again. And we don't know why. We don't know what's going on, but it's possible. I mean, blood-brain barrier is there for a reason. So if it goes awry, it's not going to be good for your brain. So it's possible that is what's driving the chronic neurodegeneration after injury.
It's great. I mean, this is TBI has always been this mystery. Like it's a diffuse injury, and for us,
right,
a lot of our work is a little more specific than that, and it's really interesting. I saw you appear, Svetlana. Do you have a question?
Yeah. Hi, Andy. We've met, talked a little bit in the past. I'm a stroke neurologist, the VA.
Right.
So what is the story then on this pathway and neuro repair and actually neuroplasticity and rehabilitation, right? So, and I'm specifically, I mean, there are two phases in neuroplasticity, neurorehabilitation, the acute phase, and that's kind of a self-repairing system that probably all these processes of neuroprotection and neurorepair are occurring spontaneously. But then in the chronic stages, right?
Right.
Can we use these pathways in rehabilitation? What do you know?
Well, the answer is I don't know, but I'm very interested in knowing. And what we are hoping is that blood levels of acetylated tau will serve as a way to give a glimpse into real time into the brain as to whether or not the nerve cells are healthy or not. So if the rehabilitation is working and the nerve cells are recovering and such, then we should see acetylated tau levels go back down to whatever is the normal range, which we haven't even established yet. If the acetylated tau were persisting to be elevated, then I would say something like diflunisal, which seems to at least potently lower it in mice and should work the same in people. that if you could give diflunisal at times like that and lower their acetylated tau back down to normal range, that might help attain functional recovery at a faster rate. That is my hope. I would love to test it.
Right, and it's also as a supportive, something that would support the learning. You know, with rehabilitation, we basically we are tapping into learning pathways, right? So if there are compounds that can support learning, that we maybe should consider looking in the animal models.
So to be honest, that was our original thought when we were looking for a pro-neurogenic drug in the hippocampus, was that it was going to be a cognition enhancer.
Yeah.
It turns out we didn't make normal mice any smarter than they were otherwise, simply by making more of those cells survive. But in all of these conditions,
smart, maybe they're too smart, but if
right. So but the tests are also designed to find deficiencies, not to show improvement over the baseline. So
yeah.
I believe that there's a robust amount of evidence that increased survival of these newborn hippocampal neurons promotes learning and promotes memory.
Yeah.
So even outside of stopping a neurodegenerative process, outside of the hippocampus, simply helping the hippocampus function better is going to help learning. And so if learning, as you say, is an important part of rehabilitation, I believe it would help. We showed, for example, that it helped. We developed a treatment-resistant model of post-traumatic stress disorder. It's a lot to go into right now. We published it last year, but basically a lot of people with PTSD, they aren't able to undergo the desensitization treatment. It just doesn't work for them. We made a model that doesn't do that. And then we showed that when we enhance production of nerve cells in the hippocampus with our compound, that the mice are able to then learn to be desensitized to the original trauma. So I believe there's a huge role for learning in rehabilitation. Not at all.
Okay. Well, that would be really interesting. We certainly have human paradigms for all of that, but it needs to be tested in animals first, probably.
Right. Well, yeah. So to give a P7C3 molecule, it has to go, of course, through FDA approval. So we're a ways off from that. But in terms of the neurodegenerative process, diflunisal is FDA approved. It's safe. If it wouldn't enhance neurogenesis, it wouldn't enhance survival. I doubt it would enhance survival of cells in the hippocampus. I really doubt that. But I do think it would stop chronic neurodegenerative processes, which would also in turn help the brain work better.
Yeah, and if it also stabilizes the already existing, so if it's promoting synaptogenesis, if it's promoting just the health of existing neurons, you know, their metabolic system, like you were saying, you know, it's all about being able to make ATP.
Right. We have shown in mice that have been genetically engineered to mimic an acetylated state that they have loss of synapses through synaptic vesicle 2 staining. And so there definitely is something to the fact that if you have too much acetylated tau, you're not having enough normal synapses. The mice become impaired. So it will just remain to be seen whether the same thing happens in people or not.
Okay.
Well, you made a statement that it's a ways off from FDA approval, and what do you need to determine still? Is it the safety or it?
It's the safety. We have to pick our. Well, for the longest time, we've had to pick what will be our clinical application, and we're still not entirely set on that because a lot goes into that because you don't want your molecule to fail in a huge clinical trial. So, and all of those areas are tough clinical trials. But the other thing is that we really want to pick our very best molecule. and then put that through all of the 2 species IND enabling toxicity studies. And that's not something that I have expertise in. So that I'm really lucky to be able to work with the Harrington Discovery Institute, because we have a miniature pharmaceutical company now essentially helping me and saying, the chemists say you should tell the Wuxi chemist to make this one, and then you should test it. And then, and they know which areas to avoid. So the help is just amazing. Before that, I had no idea what to do.
Yeah, because all along I'm thinking, well, I think this needs to be a human trial. You went through the species and, but you kind of teased a little bit, and then when you moved to the end, you had other things in clinical trials, or at least FDA approved drugs. Yeah, I'd like to. I mean, this has a lot of impact, obviously, for the VA, for TBI, and maybe for a lot of other things that
Yeah, I think so.
that we might be interested in. Other questions for Dr. Pieper? I'm going to start calling on people. I'm just kidding.
That's mean. Don't do that.
I have been known to do that occasionally, but not today.
People can email me anytime too. I mean, it's not if they want to ask questions later.
As we've talked before, Andrew, the TBI is a major problem that the VA, it's the rehab research and development service. This is a big priority, and it's something that's it's very difficult for we're mainly device related people for us to address. And I think it would be a very welcome sort of research line in the VA, as you know already, but it's something that I'm interested in trying to get going in the FES Center, some kind of a TBI activity. So maybe we can touch base again. I know things have been weird for a while here, but
I'd be delighted to team up on that. And like Svetlana was saying, it's entirely possible that this would help the rehabilitative process in many ways. And so it makes perfect sense to me to give it a try.
Yeah, cool. Okay. Last call for questions.
Do you have, so this TBI setup, you're not building a lab at the VA, are you?
We have a lab at the VA. In fact, what I showed you is at the VA.
I'm sorry, yeah. I missed that. I thought you were a case with the lab.
I have one lab at Case and one lab at the VA.
Oh, okay.
And all of the TBI stuff is done at the VA.
Oh, great.
So if anybody, and in fact, we collaborate with people all over. So we, for example, we are injuring animals and delivering them to collaborators at the Cleveland Clinic. So wherever you are, if you want TBI injured animals, we can do it.
Yeah. Well, just Bob, so we did have a TBI study. We finished it, but we had a TBI study. And enrollment, it's tough to do that. It's a difficult population to have large enrollment, even at the VA. So, but yeah.
But there's a lot of TBI related things happening in town. Obviously, what Andrew is doing. Jay Alberts does concussion related things at the clinic.
Yeah, there's more sports. There's a sports medicine related. Yeah.
This is a good application for that. I mean, how do you know when it's okay for somebody to go back and play sports? We don't really know. It's a clinical judgment. It's not perfect. Not that a blood test would be perfect either, but it would be another data point that would help people make an informed decision. And so, like if my kid suffered a TBI and their acetylated tau level was sky high, I wouldn't, and they looked fine otherwise, I would still say, hey, wait, you're not going back out there until that comes back down to normal.
And that should be important for VA and for DOD for sure. I'm sure you thought about that. Mm-hmm. Because it's the mild TBI is mostly what we see clinically, right? But that's what we see on the clinical side. That's years after probably, and but yeah, it's not only the severe. The severe is very rare.
Severe is rare, and it's
rare,
complicated
but the mild is everywhere.
Yeah, the severe is very complicated by all the hemodynamic instability and the other things that go on. The mild is more prevalent, and I think probably more amenable to treatment.
Yeah.
Okay. Anything else?
Yeah, hi, this is Ahlam.
Hello.
Yeah, hi. Maybe you actually mentioned this in your presentation and I missed it. But it seems like the compound that you're using is actually, seems from the way that you presented, is helping with a lot of neurodegenerative disease. And I was wondering what's the, like, again, you mentioned the mechanism, but I'm kind of like trying to understand. There is, even though that all these neurodegenerative disease present as axonal death, the reason why we reach that point is different, whether you're talking about Alzheimer's disease or TBI or something else. Like, for instance, for Alzheimer's disease, we're talking about like the start of the disease is different between all these conditions. So I'm really wondering like how and when in this kind of disease, this compound is helping, because it seems to me, like, I understand the signaling pathway that you presented, but I still wondering, like, each one of these diseases has different start point. So how is this compound is able to work with all of these diseases at the same time?
It is true. Each disease has a different precipitating factor, for sure. What's also interesting about each disease is you have the same genetic makeup entire life, but they don't develop it until a certain age. So there's something that changes over time that reaches some sort of tipping point where the disease starts to manifest. And that is constantly shifting as we learn more and more about the diseases. But the best reason, and first of all, I don't know the answer to your question, but the best thing that I can think of is that because we elevate NAD levels with P7C3, it's particularly helpful to cell types that are highly dependent on NAD. And so neurons are some of the most highly dependent cell types there are on NAD levels. That they have massive energy demands. And in fact, there was this spontaneous mutant of a mouse called WLDS mice that have this bizarre fusion of proteins that makes them have a higher level of NAD. And those mice are resistant to almost everything. So you cross those mice with all types of different genetic models and they do fine. So NAD is very important to neurons. And I think it's very important therefore, to helping the body fight back against this disease by helping the cells repair themselves. And so it's probably doing a downstream of NAD, it must be doing a different thing in each of those different diseases. The other cell type that is also very dependent on NAD, which I hadn't even thought about before we did this electron microscopy stuff, is the endothelial cells, but they're incredibly energetically demanding. Endothelium is actually the biggest organ in the body. I've gotten an all new appreciation for the endothelium since doing that chronic TBI study. They're very dependent on NAD. And so I think when they become energetically compromised, they start to deteriorate, and we help them that way. So I guess that's a long-winded way of saying I don't know the answer to your question, but I think it involves just making the cells better equipped to repair themselves.
Thank you.
Okay, well, Andrew, thank you so much for presenting today. It's really fascinating stuff. I'm going to be in touch because I would like to try to help get something together around town for the TBI.
For sure, that'd be great. And thank you all for listening, and thanks for giving me the opportunity to present to you today.
Okay, I'll clap. Thank you.
Okay, thanks.
Take care.
Bye-bye, everybody.
Thanks, everyone. Dr. Chen.